right, this is our Unit 14 Genetics Notes Part 2. This is going to cover Mendel's third law, which is the law of independent assortment, which is going to take us into discussing dihybrid crosses and how to do those. So we've spent some time mastering how to do monohybrid crosses. Now it's time to take that information and apply those same concepts to Mendel's third law. I've given you guided notes, so you're going to fill in the blanks as I write in the blanks on the screen, and we're going to work out an example together. Alright, so just a quick review. The law of independent assortment is Mendel's third law, and I know we've written this down more than once, but just so you have it in one more place, the law of independent assortment states that alleles are going to separate during gamete formation and are sorted independently of each other. Okay, so this means that traits are not going to be linked, right? So for example, this would mean that a tall pea plant will not always have purple flowers. Okay, so these traits are not linked together. We could have a tall plant with white flowers or a short plant with purple flowers, right? Those traits are separated independently, so we can get a mixture of different outcomes in our offspring. All right, so what is a dihybrid cross exactly? Well, a monohybrid cross was whenever we were crossing only one trait. So a dihybrid cross is a cross where we consider the inheritance of two traits at the same time. Okay, so for example, we would use a dihybrid cross if we wanted to predict the chance of having a pea plant that is both tall and has purple flowers. Okay, so we're trying to predict two traits at the same time instead of only one trait. There are certain steps that we need to follow every time we're completing a dihybrid cross to make sure that we're doing it correctly. The first step we are always going to do is we are going to determine the parent genotypes. Remember, those are our letters that represent what the genes are saying. So we're going to determine our parent genotypes if those are not given to us in the question. And I am probably not going to give this to you because we need to get in the habit of figuring those out, which I've already had some practice doing with your monohybrid crosses. All right, step 2 is that we're going to use what I call the 1, 2, 3, 4 method to determine the possible gamete combinations. So remember, on our Punnett square, we write the gametes on the outside. So when we're doing dihybrid crosses, we have to do one extra step to figure out what's going to go on the outside of that Punnett square. Step 3 is that we're going to draw a 4x4 Punnett square. So 4x4, this is actually going to end up being a total of 16 squares in size, okay? And we're going to draw this Punnett square if it's not drawn for you already, and you're going to write the gametes that we figured out in step 2 across the top and then down the left side. Right? So that's pretty easy so far. Step 4, we're simply going to fill in our dihybrid cross, and we're going to always follow the rule that we're going to group like letters together and put the capital letters first within each group. And you'll see what I'm talking about more with that rule when we actually work our example on the next slide. And then the last step we're going to do, step 5, is to determine the phenotypic ratio. We are going to determine the phenotypic ratio and in order to see what our offspring are going to end up looking like given to parents. Alright, so here's our example. Our example says, in pea plants, tall is dominant to short, and purple flowers are dominant to white flowers. It says, show the cross between two pea plants that are both heterozygous tall with purple flowers, and predict the phenotypic ratio of the offspring. Okay, so the first step is to determine our parent genotypes. Okay. So let's go back and read our question again. Look at the information given about our parents. It says, show a cross between two pea plants that are both heterozygous, and it says They are tall with purple flowers. Heterozygous, tall with purple flowers. What does heterozygous mean? You should be thinking in your head that heterozygous means different. Okay, so these are going to be different letters, meaning a capital and lowercase. And so when we look at our phenotypic ratio, we should get that tall is dominant and purple is both dominant too. So let's just start off with our first trait, which is tall. So if tall is dominant and we have a heterozygous tall plant, we know that our letters are going to be capital T, lowercase t, because they have to be different. We have to have a capital and lowercase for it to be heterozygous. Now, if we look at our purple trait, purple is also dominant, and in this case, this plant is also heterozygous for its purple flowers. So we know to be heterozygous, we're going to have a capital P and a lowercase p. So our parent genotype for our first parent would look like this. And since our question says that both parents have the same genotype, we're simply going to write this again in the next blank. So that's how you figure out genotypes. It's very similar to how we figured out genotypes from our monohybrid crosses. Now we just have 4 letters together instead of just 2. Okay, next, step 2 is to use the 1, 2, 3, 4 method to determine the gametes from our parents. So we know that our genotype we figured out was big T, big P, little p. So for our 1, 2, 3, 4 method, you're simply going to give each letter a number, 1, 2, 3, and 4. And to figure out our gametes, you're going to do a series of different combinations of matching them together. So first we're going to put 1 with 3. So when we match 1 with 3, that gives us a gamete of capital T, capital P. So that'll be our first gamete combination. Next, we're going to put 1 and 4 together. When we put 1 and 4 together, that gives us capital T, lowercase p. The next step is to put 2 and 3 together. When we put 2 and 3 together, that gives us a lowercase t and a capital P. And then the last step is to put 2 and 4 together. And when we put 2 and 4 together, that gives us a lowercase t and a lowercase p. So these are going to be our 4 possible gamete combinations. And you have to do this step before you do any dihybrid cross. So our gametes for parent 1 are going to be as follows. We just figured out. Okay, and since our parents have the same genotypes, our gametes for parent 2 are actually going to end up being the same in this case. Now, that is not always the case, so you will have to, if they're not the same, you would have to go and do the 1, 2, 3, 4 method on the other genotype as well. Okay, so let's move on now to step 4 and 5. So step 4 says we're going to write our gametes on the outside of our Punnett square and fill in our Punnett square. So here we go. So we have capital T, capital P, capital T, lowercase p, lowercase t, capital P, and lowercase t, lowercase p. So there's one parent's gametes, and then we write the other. Notice that now, instead of having only one letter above each box, now we have 2 letters above each box. Okay, if you skip step 2, you are going to mess up and not have the right things written on your Punnett square, which is going to result in the wrong answer at the end. Okay, so when we fill in our Punnett square, we're going to fill this in much like we filled it in with our monohybrid cross. Okay, same type of method. So what we're going to do in this case is we are going to start with our first box in the top left-hand corner. So in our first box, we're going to put this gamete combination with this one. Now, when we write this down, we are going to make sure that we're grouping our T's together and our P's together. And then within each group, we're going to put the capital letter first. So in our first box, we're going to get big T, big T, big P, big P. Okay, so notice how I've kept my T's together and my P's together. In our second box, we're going to get big T, big T, big P, little p, because we're crossing this box with this first gamete over here. Okay, notice that I've kept my T's together, my P's together, and then in my P's, I put my capital P before the lowercase p. Okay, and in our third box, we're going to have big T, little t, big P, big P. And in our fourth box, we're going to have big T, big P, little p. So that takes care of our first row. On our second row, we're going to be crossing. We have this gamete combination, and we're going to be simply crossing it with the one above it. So we have with the gamete at the top. So we're going to have big T, big P, little p. And then we will have our next one, we will have big T, little p, little p. Then in our third box of the second row, we're going to have big T, big P, little p. And then the fourth box of our second row, we're going to have big T, little p, little p. Right? And then we'll do the same thing with our second and fourth row. So I want you to try to pause the video and try and fill in these last two rows by yourself, and then press play when you're ready to check your answer. Alright, so you should have your dihybrid cross filled in like mine on the screen. So check your answer. And I want you to notice that in our dihybrid cross, we have 4 letters in each box instead of 2, right? Because we're crossing 2 traits at the same time. So once we have our dihybrid cross completed, now we have to do step 5 and actually complete and figure out our phenotypic ratio. What I mean by phenotypic ratio is we need to look and see how many of each phenotype we're actually going to get in our offspring. Okay, so in this case, there are going to be 4 possible phenotypes, right? We can have one that is going to be purple with tall flowers. Okay, that's going to be our first phenotype option. We could have one that is purple flowers but short. Actually, let's go back and rewrite this tall and purple, since we did our tall trait first on our dihybrid. So we'll say tall with purple, or tall white, or short purple, or short with white. Okay, so these are our 4 phenotype options. So let's look and figure out exactly what we're looking for in our Punnett square when we're trying to figure out how many of each of these we're going to see. So remember that our phenotype is determined by our genotype, and our genotype are those letters inside of our Punnett square. So tall and purple, we know that tall is dominant and purple is dominant also. So we know off the bat that if it's a dominant trait, all we need is just one capital letter in order for that trait to win. So for the plant to be tall and have purple flowers, we need it to have at least one capital T. Okay, the second letter is not going to matter, because as long as we have at least one, it's going to be tall. And we're going to need at least one capital P, because as long as it has at least one capital P, the flowers will be purple. So let's go through here and highlight or circle or mark how many we're going to have that are tall and purple. So our first box here we have both at least one capital T and one capital P. In our second box, we have at least one capital T and one capital P. Third box, we have one capital T and at least one capital P. Fourth box, we have at least one capital T and one capital P. In this fifth box, we have at least one capital T and one capital P. We're going to skip the next box because it only has little p's. In our next box, we see at least one capital T and one capital P. Box after that, again, does not have a capital P, so that one gets skipped. The first box of our third row, we have at least one capital T and one capital P. And in our second box of the third row, that one will get circled because we have at least one capital T and one capital P. The last two boxes of our third row only have little t's, so those aren't going to count. And then in our fourth row, we see that only the first one here is going to count because that one has at least one capital T and one capital P. So if we go back through here and count, let's see, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So we get 9 total that are going to be tall with purple flowers. Right? So now let's see how many are going to be tall with white flowers. So to be tall, we still have to have that at least one capital T. But the white flower is recessive. So for the recessive trait to be seen, remember you have to have both recessive alleles or two lowercase letters. So that's going to be little p, little p. So we're looking for boxes that have at least one capital T and two lowercase p's. So let's look here and see how many of those we can find. So starting in our second row, since the first row is completely, we've already circled all those. So our second box of our second row, this one will count because we have at least one capital T and two little p's. And our fourth box, the second row, will also count because we have one capital T and two little p's. It doesn't look like any in our third row are going to count because we don't have any capital T's left in that row. And in our fourth row, it looks like we're only going to have this one here. That's the only one that has one capital T and two little p's. So it looks like we're only going to have 1, 2, 3 that are going to end up being tall and white flowers out of these offspring. All right, so next we're going to look for a short plant that has purple flowers. So short is the recessive allele for the height of the plant. And so that's going to be, we know since it's recessive, we have to have both lowercase letters. So we're looking for little t. But purple is the dominant trait, so we're going to be looking for at least one capital P, and the second letter is not going to matter. So let's go through here and see how many of these we're actually going to have. So it looks like we have this one here is going to have two lowercase t's and one capital P. And so will this one, and so will this one. So it looks like we are going to end up getting 3 that are going to be short and have purple flowers. Now, to have be a short plant with white flowers, that is an all recessive thing, because short is recessive and so is white. So that means this one's going to have to be little t, little t, little p, little p. And it looks like we only have this one left down here. So that means we're only going to get one that's going to end up being short with white flowers. Okay, we call this phenotypic ratio. This is our standard phenotypic ratio for a dihybrid cross, and it is 9:3 to 3:1. And I'm going to expect for you to have that memorized. Okay, so when we have a true dihybrid cross in which both parents are heterozygous for both traits, we will always get this ratio of 9:3 to 3:1. Okay, so I hope this makes sense. If it doesn't, you might need to go back and rewatch the example. You are going to have lots of practice to make sure that we're understanding how this process works. So make sure you have your notes completed. Go back and rewatch and rewind any parts that you need to do so. And make sure that you have your notes glued in your journal and ready to be checked off.